Is it on? Yeah, yeah. Okay. Welcome everybody. We are here for public defenestration of PhD studies of Mr. Javier Galvan Freire. Welcome to everybody. As the president of the jury, I'm Alireza Valizadeh from Institute for Advanced Studies in Basic Sciences in Iran. I introduced other distinguished members of jury, Dr. Shinya Ito from Allen Institute in Seattle, USA. Welcome, Dr. Ito. And Dr. Luis Martinez from Institute of Neurosciences in Alicante, Spain. Bienvenidos. The candidate, Mr. Galvan Freire, is it fine?
It's fine, yeah.
Will defend his thesis entitled Neocortical Dynamics and Computational Mechanisms Integrating Sensory and Higher Order Inputs. This work was conducted under supervision of Professor Claudio Mirasso from IFISC, UIB, and Dr. Anton Arkhipov from the Allen Institute. Mr. Galvan Freire, you have approximately 45 minutes, up to 1 hour, to present your results and finding. After that, we have a 5 minutes break. You are free to leave the room and do anything you wish. And after the break, we have the session for the questions. I have asked to point out a protocol, an important protocol, that is not applaud. Please do not upload after presentation, because after presentation doesn't mean that the defense session is over. So keep your energy to do so after, the mark is formally delivered. So is there other thing that, so please.
Okay, thank you, Dr. Alireza. And thank you everyone for coming today, especially the ones that have come from so far away. Okay, so yeah, as he said, I'm going to present the work that I have been doing during the past 5 years of my PhD under the supervision of Claudio and Anton. So while preparing this talk, try to summarize all the work that we have done in just one sentence. I mean, it was a quite difficult task. I think that I did my best, and I think that this sentence overall summarizes what we attempt. We attempt to provide some evidence and some insights on how do bottom-up and top-down signals cooperate in the earliest stages of the visual hierarchy. To achieve this, during this talk, I'm going to cover several topics, starting from a general and brief introduction and motivation of the topic, and then I will cover the 3 main chapters, thesis, mainly the three pieces of work that we have been doing, and some closing remarks. So first, during my PhD, I have had to give some talks about my work, and I usually like to start them asking the audience this question. Are the stripes of this dress black and blue or white and gold? You may know this image because it has been shown in social media some years ago, and people enter into huge debates about which of the 2 representations was the correct one. It's quite interesting because although the image, the sensory input that every person perceives or receives is the same, they can see it in different ways depending on the inference that they do about the illumination on this image. To show you that, I take 2 pieces of the same dress and I can put 2 different backgrounds on each. If I put an autumn-like background, you may see the gold and white representation, while if I put a more spring illuminated one, you may see the other one. So it's quite interesting because although the image is the same, people can find the 2 different representations, and this depends on the contextual inference about illumination priors that people make. This leads to different perceptions. So it's a good example to illustrate how the same bottom-up signal, the same sensory input, interacts with some higher order signals about inference to generate what we call the perception of this image. But not all of the visual responses depend on these top-down inputs. We have found years ago that some of the neurons in the visual cortex respond to basic features of the sensory stimuli. So they are kind of encoding part of the visual space. For example, the experiment by Hubel and Wiesel on the cat primary visual cortex, where they see that neurons responded to bars in particular orientations. So overall, we can say that visual responses can be broadly classified into two classes. Some of them may be related to pure feature representation, just representing, as I said, particular features of the visual space, like retinotopy, orientation selectivity, motion direction selectivity. But there are other responses that require some level of contextual modulation provided by higher order inputs. Some examples are surround suppression, cross-orientation inhibition, or prediction error responses. So, as I said, our goal is to try to give some light on how this phenomena arises in the primary, or some of these phenomena arise in the visual cortex. As I said, some primary visual cortex activity reflect this stimulus feature and other more predictive and contextual influences. So to try to shed some light on these topics, we decided to follow a model-centric solution. The reason behind this is that during the past years, neuroscience have done a really good job at developing large-scale datasets characterizing the different components of the neural tissue and neural dynamics. Neuroscience have done a really good effort at characterizing the different cell types at the level of transcriptomics, morphology, and electrophysiology. Not only that, but huge efforts on microscopy, for example, in electron microscopy, have revealed how the different neurons connect at different levels of resolution. And we have pretty well measured the dynamics of the synapses. Moreover, since the development of neuropixels and other techniques, we have also been able to measure the in vivo activity of different single cells. So we have different pieces, different data that kind of speaks its own data, each of them. And each of them is incomplete in a given way. So to gain a full understanding of cortical function, what we should do is to focus on models, trying to integrate all of this information into a single predictive machine that can generate testable predictions about brain function, and also that allow us to study emergent mechanisms like synchronization or whatever other mechanism that requires a large number of cells. Indeed, this can produce a virtuous cycle where we can use all the information that we have to generate the best models that we can get, generate predictions, and see where we need data to continue to characterize the visual cortex. And this new data can be used to generate new models and indeed reinforce this cycle. So with this in mind, I jump to our first work where I focus on characterizing the cortical mechanisms behind visual flow perturbations as an example of visual response. To settle down the framework for this work, we can define what is called the predictive coding theory of the brain, which basically states that the brain is an inference machine which has an inner model of the world and continuously tries to make predictions about the incoming sensory stimuli. This framework took place at different stages in the visual cortex, where at each stage, the brain tries to generate these predictions and send it back. And these predictions are compared to the actual sensory stimuli, generated what we call prediction errors. Central to this theory are prediction error neurons, which are the ones prediction errors and respond when there is this mismatch. These prediction errors are used to update this model of the world and generate better predictions in the future. However, this is in the theoretical side. In practice, it's a little bit difficult to find these actual prediction error neurons. Although you may see a neuron responding, you cannot be sure if it is responding because of a mismatch or because of actual feature selectivity. So in 2012, the Keller lab published this paper where they mice to run freely on a spherical treadmill, and they coupled the locomotion speed of the mice to the speed of the gratings. And at some random points, they stop the visual flow of the image to generate this mismatch. Here you can see the setup. You can see the mice running freely on the treadmill, and at some random points, you can see that they stop the image to generate these prediction errors. They also measure the activity of the primary cortex visual neurons using calcium imaging, and you can see that there are a subset of neurons, particularly this one, which spikes when there is a mismatch. The mismatch is encoded in this video with the red box. You can see the activity of the neuron when the mismatch takes place. So this was a really good work. Thank you. You can see it right now pretty better. Now it comes again. Yeah, you can see there the neuron spiking to the mismatch. A couple years later, the same lab moved from calcium imaging to actual electrophysiological recordings of neurons, and they repeated the experiment. They let the mice run, and they coupled the locomotion of the mice to the visual flow of the gratings. And at some random points, they introduced halts on the visual flow. And they measured the subthreshold membrane oscillations. And they found that in layer 2-3 excitatory neurons, there were mainly two groups. Some of the neurons depolarize as a response to the perturbation, to the visual flow perturbation, while others hyperpolarize. Then they changed the experiment to generate a prediction error on the other side, and they let the mice run freely, and at some random points they showed the gratings with a speed higher than the actual speed of the mice. When they did this, the same given signs now switch signs, and the ones that were depolarizing now mainly hyperpolarize, while the ones that were hyperpolarizing now depolarize. Interestingly, when they looked at deeper layers, particularly layer 5 and 6 excitatory neurons, they saw that the response was more unimodal. Most of the neurons hyperpolarize in this case, while most depolarize. So it seems that these two layers are encoding differently, these inputs, and indeed, it seems that layer 2-3 excitatory neurons are the ones encoding these prediction errors. So they provided an hypothetic circuit for this prediction error neuron where the different subsets of layer 2 and 3 excitatory neurons incorporate the two inputs with different signs. Some of them introduce the bottom-up visual input as an excitation, while introduce an inhibition-mediated locomotion input, and the other ones the other way around. However, a year later, the Salem lab also published a work in this topic where they let the mice run freely. They show horizontal gratings going backwards, and at some random points, they post the visual perturbation. And they say that they can also see these strong responses to the perturbations, even without this coupling to locomotion. They see that the responses were actually stronger in the preferred orientation of the neurons, and that these responses were enhanced during running. So they argue that maybe these responses that the Keller lab was seeing were not actually prediction errors, but just responses for simple features in the visual stimuli enhanced by locomotion. But a year later, the Keller lab struck back, and they provided more evidence. They say, okay, if we have locomotion and the visual flow coupled and we introduce these halts in the visual flow, the response is way larger than if we just let the mice be quiet and reproduce the same visual flow. So the difference is like 10 times or 5 times. Moreover, if they do that, as I said, the difference is like 10 times. But if you just reproduce the stimuli to the mice and comparing the case where the mice is stationary with when the mice is running, the difference is just 2 times. So this factor of 2 is just explained by locomotion, and they argue that this big difference is explained by prediction errors. So as you can see, there is a huge debate on this topic, and a lot of things to be studied. And for that reason, we asked ourselves several questions. We asked ourselves how does this thalamocortical excitation interact with the recurrent circuit in the primary visual cortex to generate these two types of layer 2-3 excitatory responses even without this sensory motor coupling. And also, why does this same visual perturbation does not generate the same responses in layer 5 and 6 excitatory neurons? How does the circuit in the neocortex give rise to these two types of responses? And if these responses can be attributed to feature selectivity as the Salem lab arc. To shed some light on these questions, we decided to use what we call the Ville model, which was published by the Allen Institute 5 years ago already. It's a cortical column model representing a piece of the primary visual cortex, where they include a lot of electrophysiology and connectomics data. This model covers a radius of over 800 microns with an inner annulus represented by 400 microns radius. It contains over 200,000 leak integrate and fire neurons, particularly this GLIF3 model, and is divided by layers. Each layer contains one type of excitatory neurons and 3 types of inhibitory neurons, namely parvalbumin, somatostatin, and VIP neurons. The model also contains a thalamic module that processes visual stimuli and converts it to thalamic spikes that enter the column. And the model is overall adjusted to reproduce experimental spike trains. It's another version which includes traits, but for computational resource reasons, we decided to focus on the point neuron one. Interestingly, the model only contains one electrophysiological model for layer 2-3 excitatory neurons, which means that any difference that we see within the neurons in these layers is completely attributed to difference in connectivity, since they have the same inner dynamics, cell dynamics. So with this model on hand, we decided to reproduce the open loop experiment that the Keller lab did, particularly focusing on the case where there is no locomotion, because the model does not have a motor-like input. So we take the model, we simulated this passive viewing, and we recorded the activity of the model. These are the spikes from the LGN neurons, and this is the activity of the different layers in the primary visual cortex. As I said, we are going to focus on excitatory neurons in layer 2-3. In the previous work that I showed you, the Keller lab characterized the responses of the single neurons using fluorescence or subthreshold membrane oscillations. Given that we are using a leak integrate and fire model, membrane oscillations is not a good proxy because it is largely affected by the reset. And also, these neurons tend to have really low activity. So firing rates may not be another good approximation. For that reason, we decided to focus on postsynaptic currents, namely the total current that each neuron is receiving at each time step. And we characterize the responses measuring the variations in total input current. And we classify them using as a threshold the Rio base, which is basically the minimum current needed to elicit a spike in the neuron. So we say that the neuron hyperpolarizes with visual flow if it's below a certain threshold, and it depolarizes if it is over a given threshold. So using these measures of postsynaptic currents, we can see the two groups, the hyperpolarizing one and the depolarizing ones. You can see that the behavior is quite similar to the one found experimentally. Moreover, since we are using a model, we have a lot of information that we can extract to understand what is behind these antagonist responses. Particularly, we have the bottom-up input coming from the thalamic nuclei, the thalamic module, and the recurrent input coming from other neurons in the cortex. We can see that overall, the recurrent input tends to be inhibitory, especially for the hyperpolarizing cells, while maybe neutral for the depolarizing ones, and that the bottom-up input is largely excitatory, except for some of HbF cells which are not connected to the thalamus. Then we also wanted to check if we can reproduce also the results in deeper layers, particularly layers 5 and 6. And we saw that we also get in the model unimodal responses. Most of the neurons depolarize with the visual flow as found experimentally. Again, if we look at the different contributions, we can see that now, while the bottom-up input continues to be positive, now the recurrent input is largely positive for nearly all of them. So now that we have shown that these two different responses appear in the Ville model, try to understand what are the differences that give rise to these two types. So the first thing that we look at was thalamic connections, and we saw that the neurons that are hyperpolarized have way less connections with the LGM module, with the thalamus. Indeed, nearly over 40% of them were not connected at all with the thalamus, while DBF neurons have a larger number of connections. Also, we saw that the neurons of these two classes tend to be more probably connected and with a stronger synaptic weight. And when we looked at connections with other cell types, we saw some little differences, but statistically significant in all of the cases. For example, DBF neurons, the one that hyperpolarize, have stronger connections with layer 4 excitatory neurons and with layer 5 excitatory neurons, while the neurons that hyperpolarize tend to have stronger connections with parvalbumin neurons in layer 2, 3, and 4. But this is only half of the story, because this is just a static map of the connectivity of the model. To gain a deep understanding on what is giving rise to these two different responses, we have to go for a dynamical approach. For that reason, we defined what we called effective synaptic weights, which basically are a proxy of the postsynaptic current. For example, in this case, the presynaptic neuron spikes 5 times, so the effective synaptic weight is 5 times the given synaptic weight divided by the time window. So using this approach, we wanted to see how these effective weights change between the static and the drifting gratings period, the visual perturbation period. We characterize the activity of the presynaptic sources for the two classes. And we can see that for DBF neurons, the one that hyperpolarize, they receive a stronger current from the thalamus and from the layer 4 excitatory neurons. Moreover, they also receive increase in inhibition from parvalbumin neurons in layer 2, 3, and layer 4, which results in a no overall depolarization of these neurons. On the other hand, the neurons that hyperpolarize show no increase or nearly no increase in the current receiving from the thalamus, but they show a little increase in excitation from layer 4, which cannot overcome the inhibition that they receive from parvalbumin neurons in layer 2, 3, and layer 4. Overall, this results in a decrease in the total current that they receive, characterizing these hyperpolarizing responses. Now, we wanted to understand these different sources to see what is driving them. For example, if we focus in layer 2, 3 parvalbumin neurons, we can show that a major driver of these neurons are depolarizing with visual flow neurons in layer 2, 3. In the case of layer 4 excitatory neurons and layer 4 parvalbumin neurons, they are mainly excited by the thalamus and excitatory neurons in layers 4, also a little bit in layer 5. So now, aside from this, we have seen that these two responses appear, and we want to see if they are actually feature selective or they are encoding this visual flow perturbation. Instead of just using one direction of motion for the drifting gratings, we decided to vary it across 8 different directions. And interestingly, we find a subset of these classes of neurons that responded with the same magnitude and same sign to every direction of motion. This highlights or suggests that they are encoding this visual flow perturbation. Moreover, there was another subgroup, which was direction selective, which only responded to the perturbation if it was in their preferred motion direction. To further test this hypothesis, we perform a full field flash experiment where we show a black, white, and a black screen to the model. And we saw that the neurons that we have tagged as perturbation sensitive show really strong responses as with the same time to every change in the input, while the ones that were direction selective respond way less. So all of this study gives rise to this diagram of activity that can explain the appearance of these two cell types. So with red arrows, the excitatory contributions, and with blue one, the inhibitory ones. You can see that the thalamus excites DBF neurons and also neurons in layer 4, and this provides activity to DBF neurons, which activate parvalbumin neurons, which silence HBF neurons. The low activity of HBF neurons cannot overcome this increase in inhibition. So we can say that these mechanisms can be understood as a competition through common inhibition, where DBFs are silencing HBF neurons mediated by layer 2-3 parvalbumin neurons. So overall, we have seen that this segregation of hyperpolarizing and depolarizing responses appears in the Ville model, that the static and dynamic connectivity of the model can explain it, and also that the characteristics of the stimuli play a role in this fundamental functional division. Interestingly, we have generated some experimental predictions that can be tested. If this scheme that we have found is true, this mechanism, the activation of HBF cells should elicit or should silence the DBF cells mediated by the layer 2-3 parvalbumin neurons. Also, if we are able to inhibit the activity of layer 2-3 parvalbumin neurons, the split should stop. And if you characterize these two classes, you may found, or you should found, that they are more strongly connected between each other, and they are more probably connected. A study that came 2 years ago characterized 2 different types of layer 2-3 excitatory neurons, 2 transcriptomic classes, and they saw that negative and positive prediction errors could be biased in these 2 transcriptomic cell types. If that's the case, it could be possible also that DBF and HBF neurons are also biased into these 2 groups. So we have seen a clear example of how a model can provide some insights on cortical function. However, the predictive power of these models is not the quality of the data sets that we are using. Up to now, like every model that I know from any cortical columns introduces structural priors to generate an initial structure and connectome, and then does some manual tuning to reproduce in vivo activity. However, up to now, I haven't found any work that tries to use in vivo activity also in the construction of the model. For that reason, we jump into this work, trying to characterize a better primary visual cortex model using as priors also the functional activity, the in vivo activity. So as I said, the Ville model was published in 2020. And since then, there has been a large effort of obtaining newer and more detailed data sets about the primary visual cortex. Particularly the Allen cell types database, which we use to characterize the properties of the neurons in the model, have been extended. Now it has nearly twice of the neurons that it had at the time. Also, there has been the release of 2 major advances in connectomics, the electron microscopy connectomics from the Microns project, and the multi-patch synaptic physiology from the Campagnola group, which allow us to better characterize the structure of the model and also the connectivity information, the connection probabilities, connection weights, number of synapses, and more. As I said, there has also been the release of a visual coding neuropixels dataset, which characterizes in vivo activity of neurons, of visual cortex neurons, responding to different visual stimuli. This can allow to characterize in vivo activity, like firing rates, orientation selectivity, and synchronization, and so on. So we can use all of these structural priors to generate a model that can allow us to answer this kind of question. How does the biological structure give rise to cortical function? So we instantiated our model using all of these priors. The model can be instantiated at whatever radius size that you have, depending on your computational capabilities or resources. We took, as an example, the A400 microns model, divided into annulus and core. It contains 66,000 neurons. It nearly doubles the number of cell models that compared to that of Ville. We have increased the number of connections from the thalamus in a great effort to better characterize the thalamic axon projections into the visual cortex, into the primary visual cortex. We did that with the effort from the Allen Institute resulted in a better characterization of these projections by reconstructing 44 thalamic axons. And we saw that the number of synapses that each neurons make the number of connections that each neuron makes with the thalamus is quite larger compared to the Ville model, and also particularly for inhibitory neurons. Also, we switch from a single alpha representation of synaptic dynamics in the Ville model to 90 different double alpha synapses. Here we can see an example where we see the raw experimental data for a layer 5 excitatory neuron of the postsynaptic current, and you can see the double alpha fit and the single alpha fit. So overall, the double alpha fits give a better representation of the dynamics of the different ionic channels. However, implementing 90 different double alpha functions is completely memory prohibitive in our setup. So we decided to move to a more interesting representation using 4 alpha basis functions. So with just 4 alpha functions and weights that measure the contribution of each of them, we can reproduce the 9 double alpha functions with really good accuracy. For example, you can see here the best, the fair, the good, and the worst approach. And even the worst approach is really, really good. So this reduces massively the amount of memory and time, especially memory, that the model needs to consume. Moreover, we moved from Gaussian and uniform distributions for in degrees and number of synapses and weight distributions to log normal ones, also derived from the synaptic physiology dataset, microns dataset. And also we included a new rule for connection probabilities, which based on a recent work from the Rossi lab, where the presence of excitatory neurons is more abundant prior to the postsynaptic neuron than after it, and the other way around for inhibitory neurons. Moreover, in the Ville model, there were 2 like-to-like rules, one for connection probabilities and one for weights. Now we are only using for probabilities, and weights are uniform initialized because they are going to be tuned during training. So now I have shown all the structural priors that the model uses. But as I said, we wanted also to include in vivo activity in the construction of the model. For that goal, we used the visual coding neuropixels dataset, where they recorded activity to the presentation of this visual stimuli. And we are going to use these recordings to train some of the parameters on the model. Particularly, we're going to tune the recurrent weights of the column and the weights with the background noise. We're going to focus on just 2 stimuli, particularly the gray screen and the drifting gratings. And our goal is to reproduce the fine rate distributions at the cell type level for each of these stimuli, the distributions of orientation and direction selectivity for every cell type, the synchronization levels at the population of neurons measured by the Fano factor. And also, we want to do all of this while keeping these biorealistic synaptic weights, these log normal distributions that I showed you. Since they are derived experimentally, we want to keep them and also to maintain realistic voltage levels. So I'm not going to enter into much details about the particular components of the loss functions. They are described in the manuscript. But using this loss function that we developed, we were able to train our model. So at this point, the protocol is quite simple. We have our model, we can simulate it, we can obtain its behavior and compare it with experimental data, calculate an error, and do backpropagation. So quite easy and straightforward. However, this is totally not the case because it's not possible at all. There are 3 main problems that doesn't allow you to do this. The first one is the temporal credit assignment. It's difficult to blame each synaptic weight on the total loss and at which time. In second case, the spikes, since we are using leak integrate and fire neurons, are non-differentiable. And moreover, there are some biological constraints like Dale's law. Synapses cannot switch from excitatory to inhibitory. And also, these heavy-tailed distributions must be preserved. To solve these problems, we focus on 3 different steps. For the temporal accredit assignment problem, we switch from typical backpropagation to backpropagation through time, which properly assigns the temporal credit assignment. To solve the non-differentiability of spikes, we use surrogate gradients where the Dirac delta function, which is the derivative from the spiking activity of the neuron, is changed during backpropagation by a surrogate, a smooth function, nonlinear function. In particular, we use a triangular function that allows us to backpropagate gradients. In the third case, and probably for the first time in large-scale modeling, we use exponentiated gradient descent, a methodology that instead of using normal gradient update in the primal space, which can generate some issues. For example, if this in magnitude is larger than the actual weight, you can flip signs, and it doesn't preserve at all these heavy-tailed distributions that we see experimentally. Instead of that, we move from primal space to a dual space where we do the mirror descent update, and then we come back to the primal space. As you can see, it's an exponential update of the weights. Using all of this, we can train our model. These models have to be trained in GPUs because it takes a long time, and they are largely optimized for this. The parallel, you can parallelize all of these differential equations of the model, which are can be nearly over 1 million, and you can do it on a GPU quite fast. So we derive this scaling law of computational loss for our model. We vary the number of neurons in the model, and we wanted to see how the step time and the memory consumed increased as the number of neurons increased. Interestingly, after an initial overhead, the relationship is quite linear. If you see, the total training took over 28 hours of time, nearly over 1 day. But the most important thing is that the model runs really fast given the number and the number of neurons that it has and its size. The model can be trained can be simulated in near to biological time. Particularly, each training of step where we simulated 1 second of biological time can be run in just 3 or nearly over 3 seconds. And for simulation, it's even faster. 1 second of biological time can be simulated in just 1 second of clock time. To achieve this, it took probably over 1 year of work, including a lot of optimizations, exploding the sparsity of neurons, both in spiking and in connectivity, many other techniques. I don't want to describe them all, but mixed precision, gradient checkpointing, graph mode compilation, batch parallelism, distributed training, and so on. So now we can move from this untrained model based only on structural priors to a trained model that can reproduce all of this in vivo activity. Here you can see the spiking activity of the model, the raster plot, and you can see the orientation selectivity of the model, represented as horizontal patches of activity, and also some level of synchronization to the visual stimuli, which has a temporal frequency of 2 Hz. Here you can see the evolution of the different components of the loss function. So, as I said, now the model can reproduce all of the features that we wanted to reproduce. Particularly, we can see here the distributions of firing rates for the drifting gratings, for the gray screen, the orientation selectivity, and the direction selectivity. In each of the box plots, you can see the comparison between the untrained model, which is in light orange, the trained model, which is in orange, and in gray, the neuropixels activity. You can see nearly a perfect match in most of them. So now we have this model that has been trained and pretty well reproduces the activity of the primary visual cortex. And we want to see how it generalizes to some out of distribution stimuli. For example, if we increase the contrast of the gratings, we can see that the activity of layer 2-3 VIP neurons reduces with the contrast. And for layer 4 somatostatic neurons, the activity increases with the contrast. So there is this push-pull dynamics. Interestingly, the same dynamics has been reported experimentally. So the model can generalize to some features that has not been trained for and remains stable for every of the different contrasts. Also, we can use the model to generate or to try to gain understanding on cortical function. For example, we can answer questions like, how does the orientation and direction selectivity levels that we see arise in the model? For example, we can focus on layer 6 excitatory neurons, which has a large level of orientation selectivity. Orientation and direction selectivity in our model can arise from different sources. They can arise from the spatiotemporal bias in the thalamocortical projections. It can arise from the Rossi rule, this spatial shift of excitatory and inhibitory presynaptic sources. But it can also arise from synaptic weight tuning rules. The first two things are fixed in our model, given the structural priors, but the last one is trained. So when we examined how different neurons connect with each other in our model, we can see that two main rules appear. What we call the like-to-like rule, which means that neurons preferring the same direction of motion and the same orientation are more strongly connected with each other than the ones that prefer orthogonal stimuli. We also have the anti-like-to-like rule, which says that the connection is stronger to the orthogonal preference. And this is the case for inhibitory projections, which means that the inhibitory neuron is preventing the excitatory postsynaptic neuron from spiking in the orthogonal stimuli. We analyzed all of this and we measured the strengths of these rules using the mutual information. So we can characterize each of the connections and see where the strongest patterns of like-to-like or anti-like-to-like rules appear. We can see that the most stronger or clear connections appear in for layer 6 excitatory neurons. They form a lot of like-to-like and especially anti-like-to-like rules, which is something really relevant and that has not been pretty well characterized experimentally. So overall, we have constructed a large-scale biologically realistic model of the primary visual cortex of the mice, integrating all of this wide range of biological anatomical data sets. We have aligned the model with in vivo activity using a gradient descent-based method while maintaining anatomical realism. But probably even more important is that we have developed a scalable framework for transforming all of these multimodal, structural, and in vivo datasets into mechanistic models of cortical computation. Also, we have generated some predictions. Particularly, we have seen that layer 5 and 6 excitatory neurons receive the densest and strongest tuning modulated input, including like-to-like and anti-like-to-like patterns. That superficial layer 2-3 excitatory neurons show little tone-independent coupling, and it is nearly exclusively like-to-like. And that superficial interneuron projections show weak and even tuning modulations. So up to now, all the features that we have described are kind of purely bottom-up driven. And we also wanted to develop a framework for understanding a little bit more about higher order top-down inputs. So we decided to develop a model for the primary visual cortex and the lateromedial visual area in the mice. Why these two regions? Well, several anatomical studies and studies characterizing the functional properties of the neurons in the different areas have shown that if we try to establish a hierarchy in the visual cortex, in first place go the thalamus for sure, but then goes V1, and probably among all of the different higher order visual areas, the one that goes next in this hierarchical ladder is LM. So we decided to focus on LM. We developed a model for these two areas where to the original VLA model, we introduced a new column, smaller in size and reproducing density or a number of neurons in the actual lateromedial visual area in the mouse, and we connected them between each other. We did that using data from the Blue Brain project model. We introduce this interactive connectivity, and we also include a topographic mapping between the neurons in the model with connectivity rules derived from this connectome. For the initial interarea weights, we use the Bill et al. Intracortical weights, but it's a parameter that's going to be trained. So we trained our model to reproduce the same stimuli as in the previous work with this new B1 model. And now our trainable parameters also include the LM recurrent weights and the interarea weights between the two columns, aside from B1 recurrent weights and noise weights. We focus on the same stimuli, gray screen and drifting gratings, and we trained our model. Here is the evolution of the different components from the loss function, and here the activity of the model from LGN, B1 neurons, and LM neurons. Again, our model properly reproduces firing rate distributions at the cell type level, orientation and direction selectivity distributions, again at the cell type level, synchronization levels, this for both of the columns. Now that our model is trained, we focus on different properties of the model that the model has not been trained for, but that we know that exist in the visual cortex. For example, receptive field sizes. Receptive fields are basically the regions of visual space that elicit the most activity on a neuron. So we characterize how large these receptive fields were for each of the neurons in both columns. And we saw that there is an increase in the receptive field size from B1 to LM, as expected experimentally, which can be seen here in experimental recordings. LGN is the first, then B1, then LM. Also, with our model, to understand what is the effect of this LM top-down input into B1, we can simulate a lesion of LM and see how B1 neurons behave without the LM feedback. And we saw that the receptive field sizes slightly were cut down. So overall, we saw that LM has a receptive field 30% larger than B1, quite close to the 25% increase in the experiments, and that the LM feedback projections enlarge slightly B1 receptive fields. We also looked at another property called latency, which measures how fast a neuron responds to a stimuli. Particularly, we measure the time to first spike of neurons in the model for B1 and LM as a response to a white flash. We saw again an increase in the hierarchy with this particular metric experimentally and in our model, LGN at the bottom, then B1, then LM. Again, we simulated a lesion of LM to see how this affects B1 neurons, and we saw that B1 neurons latency was increased. So overall, we saw that the LM responses are 1.8 seconds slower than B1 responses, and then the LM feedback rate B1 responses by over 2 milliseconds. But we also wanted to study not only passive responses to visual stimuli, but also how the LM feedback can affect during a given computational task. For that reason, we focus on a purely visual task like MNIST classification, classifying the numbers. And the setup was the following. Firstly, we present 50 milliseconds of a grayscreen, then 100 milliseconds of a given number, and then 50 milliseconds of a response window. We took 10 populations of layer 5 excitatory neurons and assigned each of the numbers to one of them. And we wanted our model to make a prediction on the number that it is seen by increasing the firing rate activity of the population associated with that number. Here you can see a given example. Between 50 and 150 milliseconds, we show a number, in this case number 4, to the model. And you can see the evolution of the population activity for each of the 10 populations of layer 5 excitatory neurons. You can see an increase here on population activity associated with number 4, and in second place with number 9, which may be closest one to the number 4. So the network in this case makes the correct guess. We trained our model for 20 epochs, and we achieved a final accuracy of 93.8%. And we can see that this accuracy is more or less well distributed across all of the numbers, except maybe for number 4, which is a little bit confused by the model with number 9. But now comes the really interesting approach, and it is that in our model, we can lesion LM and remove the feedback that V1 is getting from LM. So in this way, we can get some causality on what is the role of LM in this task. If we do that and repeat the classification on the validations on the testing set, we see that the accuracy decreases by 6%, affecting mostly the representations on number 4, 5, 6, and 2. But we want to get a little bit more of understanding on how this has happened, how we lost a 6% accuracy. So the first thing that we did to gain more understanding on LM role on this task was to make the network make a decision on the actual number every 10 milliseconds. So if we do that, we see that after 40 milliseconds, the network to move upper in accuracy than the random guess, which is 10% for this task. And at the beginning, the disconnected model is the one that achieving the largest accuracies, but then it flattens out first, and the connected model achieves largest accuracies at the end, as we saw. Here you can see the dynamics at the beginning. Not having LM, having LM is prejudicial, and then it becomes something important. Also, we use multidimensional scaling to see how the network was encoding the different representations. If we do that with a connected model, we can see that the representation of the different numbers is more clearly separated in the manifold. However, if we remove LM and repeat the same, the representations become a little bit more intermingled and noisier. So it's more difficult to the model to establish clear representations of them. If we increase this feedback, not just completely removing, but step by step introducing it back, we can see that it starts to increase, and with 40% of the feedback back, you can regain nearly 50% of the feedback, of the accuracy. So if all of this makes true, if the manifolds become more intermingled, then one would expect that in noisier tasks, where the inputs are not or are less clearly represented, the presence of LM would be more beneficial. And indeed, we saw that if we start to increase the level of Gaussian noise in the image than we are showing to the model, we can see that when the task becomes more difficult, having LM, the connected one, is beneficial and makes the model more robust. So overall, we have seen that this model can reproduce spatial, temporal, and computational signatures of top-down modulation. That the model reveals an increase in the hierarchy in latency and response receptive field sizes. And the feedback of LM can provide robustness in this classification task. But probably, again, more important than this particular result is that we have developed a framework, a template for constructing and validating multi-areal cortical models under these unified structural and functional constraints. We have also made some predictions, like one should expect that the optogenetic suppression of LM feedback, especially during noisy vision, should degrade the discrimination of ambiguous stimuli, and that the LM feedback suppression should reduce both V1 responses and V1 receptive field sizes. So to conclude, I would say that during this thesis, we have attempted to shed some light on some fields, on some points in the neuroscience field. We have developed a theory-driven methodology for training biologically realistic models. We have shown how these models can provide some insights on feature selectivity versus predictive coding, this hot debate. We have seen the emergence of tune-independent connectivity in the model, and we have seen some of the roles that top-down visual inputs can have. All of this within an efficient regime, because all of this model, in principle, could be implemented in neuromorphic hardware, reducing largely the energy that they consume. And also, can be used to derive algorithms to be implemented in this hardware. Again, there are a lot of limitations, which indeed guide the roadmap of future experiments. We have not accounted for dendritic computation. We have not included neither form of plasticity. The visual stimuli was limited, and we haven't provided any locomotion-like input in the models. But if I have to give one take-home message, I would say this: like, modern neuroscience have given us with rich multimodal data sets, but large-scale modeling is the tool, is the engine that makes those parts run together, turning these static maps into a single predictive circuit. So thank you for your attention.
Thank you very much. It was very nice and informative. And we have a break for 5 minutes, and then you can come back for the questions.
Gracias. Me lo puedo quitar ya, ¿no? No, déjalo porque... Ah, vale. Vale. Creo que eso no se graba.